Hi, welcome to this edition of Snax. My name is Tim Schofield, and I'll be presenting on good graphical practices. As a brief bio, I started my career in non-clinical statistics at Merck Research Laboratories, then moved on to regulatory affairs in GlaxoSmithKline. I ended my career as a senior scientist in both MedImmune and again in GSK in their technical research and development departments. I'm currently owner and consultant of CMC Sciences and can be reached at my email, tim@cmcsciences.com, or by LinkedIn. The objectives of this particular presentation are to learn to choose the correct graphical type to both explore data, or I should say to either explore data or to communicate study results. Also, to determine the proper scale for a graph. And finally, how to annotate graphs properly to communicate study design and conclusions. Let's start with a category of graphics called exploratory graphics, which are used to address statistical assumptions such as normality of the data and for detecting outliers. Now, what most statistical methods require is that the underlying data be normally distributed, which means that most of the data populate the middle of a distribution. They're condensed into the middle levels of a response variable. However, a lot of biopharmaceutical practices, particularly bioassay, generate what are called log normal distribution, which is skewed towards higher responses. Now, a problem with this is that this can often be confused with an outlier. That is, the log normal distribution might appear to have an outlier, as seen in the middle panel, but in fact, it's a regular part of the log normal distribution, a skewed distribution, whereas an outlier is an aberrant point separated from an otherwise normal distribution. So a first thing to be aware of in using exploratory graphics is to be sensitive to the distinction between a common log normal distribution and an outlier. Now, that was good for small data sets, or it was easy to display at least for small data sets, but large data sets, which stem from model residuals, which I won't go into detail about, can be examined using more sophisticated techniques that are available in some software packages, such as normal plots, sometimes called QQ plots, or histograms. Now, if we do a normal plot, such as one I'm illustrating here that comes out of a particular software package of a log normal distribution, the fingerprint for that distribution is that this normal plot, which is meant to show data appearing approximately on the red solid line, are concave downwards, or sometimes called spilling water. And a histogram of that same data would show, as I showed in the previous slide, a skewed distribution. So these are common characteristics of data that are log normally distributed, the downward concave profile and a QQ or normal plot, or a skewed distribution as a histogram. What we would typically do with such data is perform log transformation. And we'll see once we transform the data from the previous panel, the data do fall primarily on the solid red line, a good indication of normality, and the distribution is approximately normal, or is at least symmetric. Now, in terms of choosing graphic types to illustrate the results of your study, a common choice, I'll say, almost the default choice I've seen, not only because it's the default in many software packages, but because people think in this particular way, is a bar chart. We use a bar chart, however, or I would choose to use a bar chart if I was comparing results from a categorical factor, or a factor that's not numeric or quantitative, but is simply categories like group A, B, and so forth. However, we're usually most interested not in that particular distribution, but in the extremes. So an alternative would be to rank order the bars to display the responses, showing in this case, as you might see in the upper panel, but more clearly see in the lower panel, that groups C and A give you the highest responses, if that's what you're looking for, while the other responses are much less. And in fact, the observer can detect a signal from noise, such as when we're looking at effects in design of experiments, using what software packages may include, which is a demarcation that shows which bars are significantly out spread from the rest of the bars. In this case, the rest of the bars being group E, D, and B. Now note, we could also, as we did before, use a dot plot to look at the data and determine which groups, if any, stand out from the rest. And this dot plot shows, as before, that groups C and A stand out or appear almost as an outlier from the distribution of the rest of the groups. Now, bar charts are deficient in detecting nonlinear trends over a quantitative factor. Take the case, perhaps, of dose response. Also, we collect quantitative data over a quantitative factor time, but in this case, we have a bar chart of doses. A scatter plot, however, is much more effective at looking at data over a quantitative factor, in this case, dose, in order to, for instance, determine the rate of change, which is the slope of that particular quantitative relationship between response and in this case, dose. It can also be helpful in the sense of detecting nonlinearity. That is, nonlinearity can be seen better from a line graph or a scatter plot than was the bar chart in the top panel. And while I'm not showing it here, it's also more effective at looking at what's called regression outliers. So line graphs are also more effective at displaying effects and interactions, most importantly, interactions between qualitative factors. So looking at this particular graph, we have two groups, which are bar charts, or bars in a bar chart for factors called 1x, 2x, and 3x, which may be nominal or ordinal, as it might seem in this case. Now, we can make conclusions from this particular graph, but the conclusions can be made more effectively, and the eye is not distracted by the full fill of the bars by doing a line graph instead. And what we might conclude from this is there are interactions between groups and the factor 1x, 2x, and 3x, because the lines intersect or do not have the same change in rate as you go from one level of the horizontal factor versus the two groups. These are also more effective when we're looking for differences in kinetics, in quotes, because kinetics can be dose kinetics, time kinetics, or any, if you will, trend over a quantitative factor, and use a bar chart. And this is perhaps a chart that I have the most headaches personally trying to decipher, because it's very difficult to see the behavior in the data when it's shown this way as a bar chart, as opposed to a line graph where we graph the lines associated with groups 1, 2, 3, and 4 over, in this case, is the doses 1 microgram through 4 micrograms, which again is useful for showing, if we're interested, similar patterns in the doses across groups, or as we see here, a dissimilar pattern, again, intersections between the group 4 profile and the rest of the groups. Now, one important feature of graphs is graphical scale, and I'm talking here the graphical scale of the response, or the y-axis. And this can often be used to represent a message in the data. So take two plots actually of the same data where the scale has been changed between the two plots. Now, I might ask the question, which would you pick to show sales over time? And again, if you were the salesman, I would postulate that you would choose the right one, which seems to show a more significant change in sales than the left panel. However, again, we might say, well, aren't we choosing the plot to draw the conclusion that we'd like to draw? And indeed, we may be doing that. So what we would like to do instead, and with a different graph, is scale the graph to the normal range of the measurements. And in this case, we might consider the case of potency, where potency usually ranges from, say, 80 to 120 or 125%. If we don't know what the normal scale is in the data, we might instead use the data themselves to tell us the scale by using error bars, in this case, 3 sigma error bars, to scale the data to where we usually see results, or the normal range of, in this case, the response, which is potency. Now, going back to the upper panel, if these were sales over time, the perhaps correct thing to do would be to scale that to the range of sales that's seen, say, historically, for either one salesman or for, let's call it a panel of salesmen, in order to put those plots, or that plot, I should say, into a scale that can be properly interpreted against the backdrop, in that case, of the usual range of sales among salesmen for a particular workforce. Now let's look last of all at graphical annotation. The purpose of graphical annotation is, in my mind, to highlight features of a study. So let's take an example where we're looking for increase in responses for processes 1 and 2, or 2 processes. We should use line types, symbols, or colors to distinguish factor levels. So for instance, if we're looking at process 1, we have a blue graph, and process 2, we have a red graph. However, we should be aware when plotting these that they may be copied in black and white. So we might use a different symbol or a different line type for those 2 graphs. We might include error bars to communicate conclusions. And what we might see here is that the error bars do not overlap, and therefore we might judge that process 2 is different than process 1, said otherwise is lower than process 1 in the response variable. And we might jitter these in order to be able to more clearly see the overlap or not among bars. And here I've added another factor to the plot, where not only am I looking at process 1 and 2, but I'm looking at these at 2 temperatures, 37 and 35 degrees C. Again, abiding by the principle of using different symbols and line types in order to distinguish these and be able to see the differences, even if I were to copy them into black and white. I would use effectively the horizontal axis labeling to show the study design. And so I can put the numeric values for the study design and not have to show this in text along with the table. And then I would also label the axes, including the units and the type of error bars that are displayed in the graph. With this graph, I can get around not having to give a lot of preliminary information. This graph shows most, if not all, of the information that somebody reviewing this graph would like to know about the study design and the outcome from this particular study. There are some don'ts, however. Don't use a graph when a table will do. So for instance, if you've only collected 3 results, it's a little ridiculous to create a graph of that particular study outcome. Don't use 3-dimensional plots when displaying 2 dimensions. They're both distracting, but they can also confuse the message out of the particular plot. And don't use graphical art, which also distracts from the method. Don't include things in the graph that are not significant to the conclusion that you'd like to make from the graphical presentation. So in summary, graphs are an effective way to diagnose data and to communicate the design and the results of a study. Choose the correct graphical type according to the type of data and the objective of the study. Scale responses to the normal range of the variable being displayed. And then finally, use uncertainty or error bars effectively to support the conclusion of a study. To end, I'd like to acknowledge the members of the AAPS CMC statistics community and fellow pharmaceutical scientists and regulators who have supported my passion for communication of study results. With that, I thank you.